Welcome to today's panel discussion on development strategies for current times. Current times are very special, and it's clear that over the last few years we have learned many things about immuno-oncology drug development. We're very pleased, Matthias and I, to have the panel today of very experienced and very broad understanding of drug development in the immuno-oncology space today. And we will start with brief introductions of each of the participants. I will start. I am Esteban Pombo. I am CEO of Targimmune Therapeutics and a company developing a targeted immuno-oncology approach that will go into the clinic next year. Matthias?
Yeah, thanks a lot for the intro, Esteban. Also, a warm welcome from my end. I'm also super thrilled on the discussion today. I think we'll have really action-packed 55 minutes. As someone already said, so my name is Matthias Nürnberg. I'm heading Global Business Development and Alliance Management at Merck KGaA, Darmstadt, Germany. So maybe just as a brief introduction for those of you that are not familiar with Merck, Merck is a 350-year-old company really operating in healthcare, life sciences, and electronics. We have an annual revenue of roughly 17.5 billion euro, of which we reinvest in R&D roughly 2.3 billion as of 2020. And of that, roughly 1.6 billion go into healthcare R&D. The areas of focus for us are really oncology, immuno-oncology, immunology, fertility, endocrinology, and cardiometabolic diseases. And when we think about partnering, really, we currently look at the full spectrum from early to late stage innovation. Three things that are important for us here may come obvious to the mind, but just to reiterate them, it's really commercial potential, synergies with our portfolio and expertise fit. So for us, it's really important that we can make a contribution to our partner's success as well and create win-win situations. When it comes to overall partnering modalities, we think of it really in three dimensions. One is personalized deal making, second is speed, and third is size. This is not something people may automatically think about when they think about Merck KGaA, but at the end, we feel more like an established biotech than really a smaller, large pharma player, where we really have an understanding for opportunity cost and this personalized deal making to generate a win solution for everyone. So if you're interested to learn more, please really reach out to me or my team via LinkedIn, or also visit our partnering website at merckgroup.com. So looking forward to the panel discussion, and thanks a lot for the attention.
Thanks very much, Matthias. If we could continue with Aram, please.
Thank you, Esteban. So Aram Mangasarian here. I'm CEO of Noxon Pharma. So Noxon targets the tumor microenvironment by directly neutralizing chemokines like CXCL12 or CCL2. And these are involved in a number of aspects of cancer biology, including exclusion of immune effector cells from tumors, recruitment of immunosuppressive cells like TAMs and MDSCs, and even repair of damaged blood vessels by vasculogenesis. Now, one of the complexities of developing agents like these is that they really need to be developed in combination. So as an example, we've got an ongoing trial right now in first-line glioblastoma patients, LMGMT unmethylated, so this roughly half of the first line that won't respond well to chemo. And we're treating them with NOX812, our anti-CXCL12 agent, and radiotherapy. So we know from animal models that the agent alone has no effect without chemo, without radiotherapy. But with radiotherapy, we have 100% complete response. So we've of course gotten the comment over the years that agents without monotherapy activity are a lower priority from investors and pharma companies, which I have to say puzzles me a bit if we're trying to make the TME a more important part of treating cancer. By the way, data from our trial is pretty encouraging. The low dose of A12 actually showed tumor shrinkage in all 3 of the first patients, and 1 of them had a durable objective response. So this is the type of thing we're targeting. Some of the issues we're confronted with in developing these kind of drugs are how do you dissect the individual contribution of different drugs when you're treating with a combination? How do you work out the downstream strategy, especially if you're layering combinations on top of chemo later, and you're starting from a single arm trial where you need to benchmark your activity. And then things like convincing people that overall survival, which we saw quite interesting effects potentially in a pancreas cancer trial, combining our agent with PD-1s, even though we don't have objective responses, that if you get 1 to 2 years survival in 4th line, that should be interesting.
Great.
So I'm looking forward to talking about all that with everyone today.
Super, Aram. Thank you very much. If I can pass on to Richard, please, if you can tell us about your experience in Bergen Bio briefly.
Thank you, Esteban, and thank you for the invitation to be here and participate in this panel. So my name is Richard Godfrey, the CEO of Bergen Bio. We're a biotech company developing agents against receptor tyrosine kinase AXL. We're really the first to understand that AXL mediates aggressive traits in many cancers. Aggressive cancers, of course, are ones that no longer respond to chemo, that are able to evade and avoid the immune system, and of course metastasize. More recently, we've learned that AXL also has an important role to play in fibrosis, and interestingly, AXL also facilitates viral entry, and we have 2 trials that are now completed recruitment in COVID-19. So very, very, very important receptor, tyrosine kinase, AXL, that mediates aggressive diseases. We have 2 therapeutics against AXL, a 1-a-day, highly selective, potent pill called bemcentinib, and a fully humanized, functionally blocking antibody called tilvestamab. We're completing some phase 2 studies with bemcentinib, both monotherapy and combination with chemo, and also combination with immune checkpoint inhibitors in COVID, leukemia, and non-small cell lung cancer. And I really echo the comments made earlier about combination therapy versus monotherapy, and endpoint determination in the preparation of therapies for the future for patient benefit. And I know we're going to talk about that during the panel. So thank you very much. Great pleasure to be here. Looking forward to the conversation.
Thank you, Richard. If I can pass on the baton to Ray, please.
Thank you. My name's Ray Takegiku. I'm the founder and CEO of Bexion Pharmaceuticals. Bexion is a company that's focused on leveraging what we believe to be an underappreciated therapeutic target, meaning the lysosome. Beyond the known origin of the organelle as the trash bin or cell recycler, the lysosome is being increasingly recognized as a pivotal meta-orchestrator of cell homeostasis. And so our origins were really in lysosomal storage diseases, but we are now known to be a clinical stage oncology company with phase 2 programs brewing in brain cancers and newly diagnosed colorectal cancer. These are, of course, diseases where the lysosome plays important roles in cell proliferation and malignancy. Moreover, our focus on the lysosome has brought us into the CNS space with a particular interest in treating peripheral neuropathies. And we also have preclinical programs in Parkinson's and other CNS diseases. At Bexion, we're really excited about the multiple therapeutic opportunities presented by targeting the lysosome. And I, like everyone else, really appreciate being on the panel and being able to, hopefully I can contribute.
Wonderful, Ray. I'm certain you will. Peter, can you give us a brief introduction?
Sure. My name's Peter Llewellyn-Davies.
Thank you.
I'm CEO of Apeiron Biologics. And as you can hear from my accent and my name, we're based in Vienna, Austria, which is evident on the picture behind us. Our company has been around for a while. We've been quite successful in developing immuno-oncology drugs. In particular, one was approved for the treatment of pediatric neuroblastoma in the EU and brings us nice revenues. Additionally, we have a very innovative autologous and transient cell therapy approach, which is entering a phase 1b trial. We've already proven in previous trials that we've shown for solid tumors in particular, like colorectal cancer, metastatic pancreatic cancer, and others, that we have a 28% stable disease over many years of some of the patients who are late-stage cancer treatment. So that is our main focus, but we also have respiratory disease treatment through ACE2, recombinant humanized ACE2, which we're using and have completed successfully a COVID-19 trial, and are now entering further trials in the next few months to treat mid and moderate cases, mild and moderate cases of the disease. So thank you very much for the opportunity. We've had many challenges in our clinical trials and development over the past years, and looking forward to sharing our experiences with the panel.
Great, Peter, thank you very much. Jürgen, your turn.
Yes, thanks. So I think also, as mentioned already by the others, I'm also pleased to be attending the panel today, and I'm really looking forward to the exciting discussions with my colleagues. So I'm Jürgen Garmer, heading Business Development and Alliance Management at Apogenics. And the company has developed innovative immuno-oncology therapeutics for the treatment of cancer and viral diseases, targeting especially one protein family, and that's the TNF superfamily, to restore a proper immune response in these diseases. Now, our lead candidate called Asunercept is a CD95 ligand inhibitor and has demonstrated a statistically significant efficacy in a controlled phase 2 study in recurrent glioblastoma. It has an overall survival benefit and a statistically significant PFS benefits. And we also have positive efficacy data from phase 1 study in myelodysplastic syndrome. Now, based on the convincing scientific rationale as the CD95 pathway plays also a role, that was where it actually was discovered in the viral infection diseases. We have started a phase 2 study in COVID-19, which is currently recruiting. And so we think that we will have the final data available until autumn or end of this year. I'm attending this panel as we are developing a new protein format that stimulates actually the TNF superfamily receptors, such as CD40, CD27 GITR, which are attractive targets to actually stimulate the antigen-presenting cells and also the T cell response against the tumor. These so-called HERA ligands make use of the natural trimeric ligand that is required for proper receptor stimulation. And the use of this natural ligand has clear advantages over other agonistic principles like using antibodies. Now, clinical data achieved with agonistic antibodies have shown that actually they have a very limited therapeutic window due to the mechanism action, because antibodies can't create the required trimeric receptors on the surface. Now, these antibodies show either a trend in efficacy, but this comes along with severe safety issues, or they have a good safety signal, but then combined with poor or no efficacy. So at the end, the challenge is that agonistic molecules, especially antibodies, to stimulate these receptors have a very limited therapeutic window. Now, and that is where we do see an attractiveness in our approach, as we are using the natural ligand and stimulating a proper signaling. These molecules come with proven less toxicity combined actually with the superior efficacy. And that's actually, of course, what you want. And for the first time, these receptor agonists can be used to have a fairly broad therapeutic window, allowing much more flexibility in the dosing in clinical studies. So the front runner here has already been licensed to AbbVie, and they just recently have started a second phase 1 study in multiple myeloma, along with an ongoing study in solid tumors. The platform is versatile, so especially the bispecific format we have generated meanwhile, combining the ligand with any targeting moiety. So we are looking for partners that really help us to exploit these versatile platforms. And as I said, I'm excited to hear and also to contribute to the questions that will come up through the discussions. And yeah, happy to participate here.
Thanks very much, Jürgen. Scott, please.
Thank you, Stefan. Thank you very much for inviting me to this panel. I look forward to the discussions. It's going to be great. I'm the Executive Vice President of Development at Nanobiotix. Nanobiotix is a biotechnology company. It has an office in Cambridge, Massachusetts, but our headquarters is in Paris, France. And it's a unique company focused on oncology and curing cancer through physics. Our lead product is NBTXR3, which is a novel, actually first-in-class radio enhancer composed of functionalized hafnium oxide crystals, nanoparticles. It's administered directly into a solid tumors, whereby it's inert until it's activated by radiotherapy. It has been designed to increase the radiotherapy energy deposit inside the tumor cells, thus increasing the cellular death, and compared to radiotherapy alone. It also protects the healthy tissues around it because it is a focalized increase in cellular death right around the nanoparticles themselves. The physical and universal mechanisms of actions of NBTXR3 is designed to trigger cellular destruction, and most importantly for this conversation in this panel is primes an adaptive immune response. And currently, similar to everybody on this panel, we have an ongoing trial. This one is in the United States in the middle of the pandemic, and look forward to talking about the impact of the pandemic on conducting a clinical trial, especially in immunotherapies. But our phase 1 trial is looking at the combination of radiotherapy, NBTXR3, plus an immuno-oncology agent in kind of a basket trial design. We're looking at head and neck cancer, but we're also looking at patients that have metastases in the lung and the liver that are being able to be injected with NBTXR3 and then followed by radiation and then treatment with immunotherapy. What's unique about this trial is that we're looking at patients that have both failed an immunotherapy, a prior immunotherapy, so a non-responder, and seeing if we can convert them to responders. And then we're also looking at naive patients and the impact. We're also having the chance to evaluate the immune stimulatory response. We know that radiation therapy alone has an immune stimulatory response. We know NBTXR3 can enhance that. And so we're looking also at lesions that have not been injected with NBTXR3, have not received radiotherapy, and seeing if we have a systemic response or an abscopal response. So, a broad program, the mechanism of action allows for tumor agnostic, whether it's with NBTXR3 plus radiotherapy alone and or in combination with immunotherapy. So like several others here, really interesting technology, novel technology, but also developing this in the time of a pandemic also has its own concerns and issues that we have to address. And I too look forward to our discussion today. So thank you very much.
Great, Scott. Thank you very much. As a audience will have heard, really, we have a broad variety of mechanistic approaches that address the immune system and the immune response against cancer in many different ways. And yet the need to develop these drugs in an efficient way and demonstrate efficacy and patient benefit is common to everybody. So we will have some common themes, and we will have some different aspects and different ways of addressing these themes, which are relevant for everybody. The other element is important in terms of the combination approaches that we have heard. That is something that is a very strong theme in the immuno-oncology space. Not only that. So let's, looking at really the last 5 years or so of drug development in the oncology space, there have been very significant changes, and some new drugs have been approached, some new patient selection mechanisms, and of course, the different strategies have been tested, some successfully, and some have been lessons that probably should not be repeated. So less successfully, if we go to that. And for that, I'd like to start with Richard and ask him, which of these strategies have stood up for you in the last 5 years or so, and why, what are good aspects, good elements, or what are bad or limitations or shortcomings of these strategies? Richard.
Thank you. Yes, I think you're absolutely right. Of course, the last 5 years has seen quite a transformation, in particularly in oncology, in the way in which we're now treating the patients primarily or ideally with an immune promoting agent that promotes our immune system to clear and control the disease. Some of the thoughts I have around this dilemma, of course, is that by and large, we're still using the old-fashioned chemo performance measurements to evaluate a therapeutic, the so-called RECIST measurements for tumor shrinkage or stable disease, the CR, the PR, the stable disease. And that doesn't necessarily always fit, and it certainly doesn't necessarily fit with what the patient would ideally like. Of course, to see the tumor shrink is a really nice observation as a patient, but cancer is such a dreadful disease. Patients are really primarily concerned with what's the survival horizon, what's the outlook for the patients, and certainly it's quite a good surrogate. Seeing tumor shrinkage is quite a good surrogate. Seeing complete response is quite a good surrogate for survival, particularly in the first line. But when you move into the second line setting or the relapse, or indeed maybe the refractory setting, CR doesn't necessarily act as a good surrogate for survival. Non-progressive disease actually is a very good outcome for patients. Seeing the disease is not getting any worse, they've stabilized, their quality of life maybe has improved, and often that will materialize in a survival benefit. So I think there's a challenge there for the industry to catch up with the ways in which we're treating cancer. One of the paradigms that I'm trying to champion is that AXL, the tyrosine kinase that we're working with, actually is a survival program harnessed by cancers. In other words, its fundamental mechanism is to allow the cancers to survive. And by inhibiting cancers, you don't kill the cancers, but you stop them from surviving. And therefore, they are tractable by the immune system. They're more sensitive to other drugs, and maybe they don't metastasize. Or if they do metastasize, they don't seed and germ secondary tumors. And this is a really good outcome for patients. So I would say that thinking about the endpoints and not necessarily being too skeptical of a drug that doesn't necessarily have RECIST performance, particularly as a monotherapy, is a way by which we could open our minds to maybe more smarter ways of developing medicines in the future. Patient benefit, primarily survival benefit, should really be front of mind. If I could just add one more comment whilst I have the microphone, and that's also use of companion diagnostics and selecting patients that are most likely to benefit. Again, that's becoming more and more sophisticated in molecular diagnosis, understanding how we can identify and target the right patients, either with a single marker or a signature of response. That is something else that I think has seen quite a lot of favor, but it's kind of ebbed and waned, I think, a little bit in terms of its utility. Clearly, the driver mutations are a good case study, but that's often just a single point diagnosis. So that's an important development that I still think has got still some distance to run. And in part, I think it's really important that we're able to secure serial biopsies. So just getting the single pre-treatment biopsy is often quite challenging, but being able to secure serial biopsies during the treatment, particularly in early drug development, is so informative about your on-target activity and what's actually happening within the tumor and the tumor microenvironment. So that's probably the second element that I'd say we could really learn from. Not necessarily just looking for tumor shrinkage, and definitely looking for the serial biopsies and seeing the longitudinal effect of your treatment on the tumor over time. They're two areas that I think we could really learn from and try and champion them going forward. And I believe that would facilitate more effective medicines that provide greater outcomes and hopefully fewer side effects and more tolerable treatment regimes.
So one important aspect is the science-driven clinical trial based on biology that you can observe from the biopsies, and that is critical. Ray, what is your views on this?
I absolutely agree with everything that Richard just said, particularly in the recurrent setting. While you would like to see a CR or a PR, stable disease is incredibly important. Richard mentioned RECIST. I mean, that's also true for RANO, right? There are limitations to the value of these imaging technologies. They're static pictures in time. And Aaron mentioned the microenvironment, particularly with regards to these immunotherapies. It's not necessarily what's happening to the tumor, it's what's happening around the tumor, to the tumor. And so, phenomena like pseudoprogression have come into the forefront. The imaging modalities that are accepted don't necessarily reflect biology, right? So PET imaging would be one in which it does, but the utility of PET and the acceptance of PET by regulatory bodies is not universally accepted. So there's been mention of biomarkers as well. In addition, some tumors are we're not able to get serial biopsies, but the availability of biomarkers that are diagnostic of what's happening at the tumor, what's happening with immune cells around the tumor, I think become important, and they can become, I think, surrogate endpoints. It's just obviously the regulatory authorities are always going to be delayed with regards to the science, and it's up to us to kind of make the science real and valuable for everybody. But I would like to comment on PRs and CRs. I mean, with targeted therapies, we've become accustomed, and indeed the world sort of desires PRs and CRs, but for the patient themselves, obviously seeing that would be a good thing. What's more important is to have that lasting, right? And what we've come to find out with targeted therapies in particular, even with the origin of chemo, is that tumors are heterogeneous and they will come back. So even with a PR or CR, it's how durable is that response? And so these are all important contributors to how we look at the disease and how we treat the disease. And yet we're sort of bound as drug development organizations to conform to RECIST and RANO. And obviously the regulatory authorities would like to see overall survival. We all want to see overall survival. That becomes rather difficult for some of these tumors in which, that are not acutely fatal. For those patients that are going to survive on standard of care for 5, 6, 7 years, those are still diseases that are in need of better therapies, but to ask a small biotech to run a clinical trial that is going to cover that many years is just not realistic. So, we really have to find a better way to describe tumors.
Yeah, so the approach of developing new ways of reading results from clinical trials that are more relevant is, of course, important. Matthias, you had a comment?
Yeah, absolutely. Despite the fact that I of course, agree to whatever was said already here by Ray and Charles, I think one of the things where I want to put a little bit of a spotlight to is really the trend of the last years that we saw on maintenance treatments in IO. So that really not looking at late stage lines of therapy, but really early lines of therapy. And just for those who are not familiar with that concept, the basic idea is to not really start off with a combination treatment straight away, but really to rather switch from an induction therapy, partly chemotherapy based, to, for example, a maintenance treatment with a checkpoint inhibitor. And just really to remind us that there were positive outcomes. So there was a specific study with an OS HR of 0.68. Yeah? So durvalumab in stage 3 NSCLC. We had the SOLO-1 study, olaparib in ovarian cancer, PFS HR of 0.3, and also the Javelin Bladder 100 study where we tested Bavencio, our PD-L1 antibody, in first-line MUC with an OS-HR at 0.69. And I think especially in settings where disease is super aggressive, that may be NSCLC, it may be UC, it may be others, this opens up a very interesting, let's say, avenue beyond, so to say, the pure OS and PFS values that we look at, because at the end, more patients can benefit from potential upsides of, let's say, IO treatments. And that goes also back to a comment that was made before. At the end, we don't select necessarily patients on biomarkers, but really on the outcome of the initial treatment. And I think that is a paradigm shift I believe we will see more and more also in the overall treatment paradigm, where we think really along the continuum of care, how to combine, let's say, established standard of cares in the clinic with novel modalities. And I think that goes very nicely also with the discussion on, let's say, thinking about what, are the endpoints that we currently accept as the standard, are these the right ones in this setting, or should we really rethink a little bit what benefit at the end and means? And I'm looking forward to further explore that.
So we've heard a lot about the deficiencies of existing clinical models and how they are perhaps in need for improvement. But if you have a molecule and you have a project and you have to move it forward with the best way forward, what are the kinds of things that would influence the choice of indication or the choice of trial design? You can go for a focused, very narrow population where you have high probability of success of a mechanistic driven trial, but not really, so the medical need may be limited, but you may have something that you say, well, no, I like glioblastoma, just to say that, because many people in the panel are actually very bravely developing drugs for glioblastoma, where the medical need is enormous. And yet the success in glioblastoma is significantly less than in breast cancer, for example. And so what are the factors that influence your development strategy or should influence the development strategy? Jürgen.
I think there are various factors influencing the development strategy. Of course, if you've picked a certain indications like we have done with glioblastoma, of course, you need to look at what kind of treatment phase you'd like to enter. Is it newly diagnosed glioblastoma? Would it be recurrent disease? And usually people start with a recurrent disease. And in this disease, you are confronted with a situation which is true for many late-stage cancer patient population. What is the actual, especially if you'd like to perform a randomized control study, what is actually the comparator? So what is the standard of care if there is no standard of care? Of course, the regulators, they come up with, best supportive care. So just take anything, which makes it really hard to find actually what you'd like to achieve, to have a defined patient population where you see a very clear population giving a randomization process where you have your treatment compared to the control arm. If you have best supportive care, you have possibly a mixture in glioblastoma. It's lomustine, it's radiotherapy, it's whatever you can think of. It's even clinical trials. You have a huge mixture in your control arm, which makes it nearly impossible to have a very defined comparison between the two arms. So that definitely what makes it complicated. So of course, so it's better if you have an indication or if you have a patient population where you have already a clear defined control arm you can compare with. Also, clear endpoints make it easier. Just a few, I completely agree with Richard and the others commenting on the endpoints, actually, especially if the endpoints drive the decision-making throughout the process. If, let's say, the endpoint is progression-free survival or overall response rates, and the decision is when the patient has achieved progression, you have to take him off therapy. So that's, of course, you're based on an unsecure endpoint. You make a decision which is probably not even good for the patient, and which, of course, mixes your result in the clinical study. So this is also something that you probably need to consider. Recruitment speed. Have you access to the patients, or are you confronted with a patient population that is really hard to recruit in the current situation? And of course, I think no need to say that medical need, competitive situation also need to be considered. So it's a really multiplex decision that is required, leaving out scientific rationale, of course, and all the other kind of data that you have generated that guide you to this decision. So complex decision, and looking at the trial design, of course, what there. It's not so new anymore, but adaptive trials are adding to the complexity. I think, Aram, we have discussed throughout the presentation here, preparation of the presentation, the glioblastoma study in the US that is actually a really innovative but complex with adaptive design, basket studies, umbrella studies. All these are designs that will be growing but are, I would say, sometimes complex to do because they even, let's say, make the comparison between the various arms even more complex.
Thank you. So some cautionary tale there, and obviously some pretty good advice. Aram, please.
Thank you, Esteban. Yeah, Jürgen, I find a number of things quite attractive about some of these adaptive trials, and without going into too many specifics, because we've looked at a few and a couple indications, but one of the things is that it allows you to explore multiple lines of treatment at the same time. And then some of them have actually are quite clever, and they'll say, all right, you're going to graduate from phase 2 to 3. And then if you're doing better in one line versus the other, we're going to, rebalance recruitment. And so the pivotal part of the trial will only be in whatever first or second line. And there also, I think, you get there's a lot of setup from what I've seen in some of the better ones, where there's heavy regulatory interactions. And you get a lot of input from the FDA, in particular for the US-based ones or the US initiated ones, if I can put it like that. Some of them have ambitions to go to Europe and even China as well. And I think that's quite exciting if they're able to manage that, because my sense is it takes quite a long time. And that would also be limiting for a small company to be able to spend that time and money and have those negotiations. I'm curious if you know, what your take is on that as well.
Yeah, agreed, Aram. So I think on the so actually we also have the trials that we are currently running, that we are planning. The GBM trial has an adaptive design built in, exactly for that reason, to really build on the knowledge that you gather in the phase 2 and transfer that after the decision-making process into the phase 3 part. And similarly, also the COVID study has an adaptive design where we have used a broad range of dosing scheme that we are testing in the first runs, and we are actually seeing the first data coming in, which in the first interim analysis look quite exciting. And we are now meanwhile focusing on one dose which provided the best results, and we take this dose compared with the comparator arm into the next stage. So yes, indeed, I agree. Adaptive trials do have advantages and are attractive. You need to do a bit more statistical analysis. You need to do a bit more on the design side, but you win if everything is well designed. You will win a lot throughout transition from phase 2 to phase 3 or at whatever stage you are. Yeah.
Yeah. So now we move perhaps to other considerations. Matthias, can you follow up with the next questions?
Sure, absolutely. So I think we touched already a little bit on the topic of patient centricity. So let's maybe dive a little bit into that. I guess the question that I have is, what's the role really or what role I should better say, patient organizations and patient experience play really in designing our development strategies? Maybe Scott, do you want to comment on that?
Yeah, thank you very much, Matthias. For me, I've had the advantage of being able to sit down and talk to a lot of the patients that take part in my clinical trials. And I'm always asking them what's important to them. And, we've talked about, Richard just bringing up, of course, overall survival, duration of a CR, a duration of a PR. We're kind of fixed in our development because of what the regulatory agencies around the world want to see. But for the patients I've talked to, whether it's my oncology patients, whether, depending on what the oncology tumor type is itself, but ALS, Alzheimer's, I've been fortunate enough to talk to a lot of patients. And then, of course, my own dad, who passed away from glioblastoma. They want quality of life, too. They don't want to live an extended 2 or 3 or 4 months and be in a vegetative state, or not being able to play with their grandkids or anything like that. And we miss that in our drug development. And I think we're forced upon missing that as a focus point because of what our regulatory obligations are. And so speaking to those patients, speaking to the patient advocacy groups and giving them insight, or having them give us insight in what's important to them and as patients, and whether it's the patient journey during a clinical trial, we've talked about that in our pre-meeting of the burden that's placed on them. We can gain a lot of insights about what's important to them in thinking about what outcome measures we want to look at, what do we want to focus on, but more importantly, when we're doing a clinical trial and we're asking these patients to participate, it's hard for them to understand, stable disease is a great thing, or CR, we're looking for the survival. They want to hear about what it's going to benefit them from a quality of life. What's the potential, right? Am I going to be able to take care of myself? Am I going to be able to swallow better? Am I going to be able to do this or X, Y, and Z? That gets them interested into a trial. And I think, the patient advocacy groups also have that inward ability to talk to a lot of patients and bring them on board. But we need to bring them on board because they want to see a benefit to themselves and eventually, quite honestly, to their family unit. And we need to do a better job. And I think I'm saying this, we as the industry, of being able to bring this to the regulatory agencies. We all know on this panel that the FDA is less associated with quality of life than Europe and some of the other regions of the world that we've worked in. But if you have no impact on quality of life and you increase survival by 1 month, is a patient really going to care? And I think we really need to take that into account as we design our, think about what our development plan is and what our endpoints are really going to show and what's meaningful to the patient themselves and also to their family.
Thanks, Scott. I personally do really very much appreciate your perspective here. And I also believe that the more and more cancer becomes a chronic disease and the more and more we discover treatments that allow us to treat patients on a more continuous basis and relatively low side effects in oncology, which unfortunately is not yet there for all indications, for all patient populations, we know that. But the more that stage, is coming closer, the more I hope also the regulators open up, so to say, their perspectives to that. Maybe Ray, do you have a perspective on that topic?
Yeah, I echo everything Scott just said, as I'm sure everybody on this panel would. I do see a rise of patient advocacy, right? And I think the FDA here in this country definitely will pay attention. They will listen. And this quality of life thing, I think, is becoming ever increasingly important, as opposed to feeling sick and maybe living, a couple of weeks longer. The trade-off is just, I think most people would not take that trade-off. So I think the regulatory agencies are appreciating that. I think the problem is, it's not necessarily a scientific thing. Right. It's a qualitative thing, and so it's an emotional thing. And so the extent to which that can sort of become more scientific, I guess the way that can be more standardized, the more it will be accepted. One of the reasons that Bexion is in peripheral neuropathy is because we had some patients who experienced benefit in our phase 1 trial. It was, of course, a recurrent population. Some patients had severe chemotherapy-induced peripheral neuropathy, and while maybe they got off of our drug because of this progression thing that happens, they did experience benefit in peripheral neuropathy, and that made a huge difference for them. Mm-hmm. The fact that they could button up their shirts, they could tie their shoes, they didn't need a walker. They thanked their PI for that benefit, and so this whole area of needing to look at the whole patient, I think, is an obligation for all of us, and is an obligation of regulatory authorities to pay attention to it. In another vein, I think patient advocacy organizations have played a huge role in rare diseases, right? Obviously, they have influenced Congress here in this country for incentives to work in that area. And they are also an important organization for getting patients into trials. So, they play a multitude of roles here. And so I think they're an important part of our whole drug development, drug approval world.
And especially in areas where they don't have major hospital institutions, in those parts of any country. The patient advocacy groups are one of the only places those patients have to go to find out about clinical trials or how to get involved in clinical trials. So they have an outreach far greater than, we do as companies. Mm-hmm. And I think we need to take advantage of that also, Ray. I fully agree with you.
A very good discussion here. Maybe to dive into one of the points, and I think Scott, you said it already a little bit, differences how, for example, the EMA, the FDA look at certain topics, endpoints, etc. Maybe just to pose the question, I mean, is there potentially a difference strategically, really, on where we conduct our trials at the end? US versus EU versus other countries. Peter, do you have a perspective on that?
Yeah, sure. I mean, let's not forget, most of the money provided for biotech companies is available in the US and not in Europe. So some of the investors who want to actually provide funding for trials say, or let's say they recommend that the funding comes from the US and through an FDA approved and FDA completed trial. So this would mean that most European companies specifically look to go to the US and get the trials completed there, quite simply because they know that it's easier to get US funding. That's a general case. However, the strange thing is that when one doesn't actually do an FDA trial, it's more difficult in particular for European companies who just did a European trial to go to the US and say, look, these are our results. What do you think? And if we speak to the FDA about this and they say, well, great, how about doing some trials in the US? And I think personally, there is a bias towards European companies, quite simply based on the experience we've had so far and how easy it was to get something approved in the in the EU compared to getting it approved in the US. And we've found US companies with less patients, with less efficacy data to getting their approval ahead of us. So that's a very strange notion we've experienced.
Thanks for that comment. Jürgen, what's your take on that from a European perspective?
I think not surprisingly, having seen that, I don't usually talking about biotech. You want to leave out politics, but it's also that's the environment. I agree with Peter that of course the US is dominating in providing the financial resources. The stock market is much more lively compared to whatever is available in Europe, and also this, of course, creates a much more lively, also in terms of deal making, a much more lively environment for biotech companies. So for sure, it will be easier if you are living in this environment, and if you are doing your trials, you are connected to the key opinion leaders. If you talk to other parties, being it investors, being it pharma partners, of course, a lot of them work with. Usually, everybody's doing it. You ask your friend, what do you think about it? And of course, they also work a lot with key opinion leaders. So you need to be connected to the right person and need to have a network. So, and that's also far easier if you are connecting in the US, if you are gathering your key opinion leaders. Again, if I think of glioblastoma, you have the typical dominating key opinion leaders. You have them in the US. And if your compound is not known in the US because you have never done a trial there, you have not recruited a single patient there, this will create, this will give you a hard time to convince US investors, to convince US pharma companies, biotech companies to really engage in a partnership. And of course, the only advice there are many more parameters you have to consider. But if everything is equal, given these reasons, I would at least advise to reach out to the US as soon as possible. Of course, it takes time. It comes with resources and so on and so forth. But at least whatever you can do from Europe. Having a footprint in the US would be advantageous.
Yeah. I think that's a very good summary of that discussion point. Esteban, do you maybe want to close us off?
Thank you. Yes, I'd like to probably go back to one of the key questions, which is really in terms of this, the experience through the COVID days. People are talking about this has been perhaps the most unusual period of our lives. And none of us has been through this massive global disruption before. And getting used to new ways of doing things and trying to be creative and make sure the projects advance. And we can speak to the patients, we can speak to the clinical sites, and the supply chains are interrupted. Really, I mean, it has been an extraordinarily difficult time. And this has also spawned a lot of innovation and creative ways of moving things forward. Of course, we all see what is possible with vaccines when you have hundreds of thousands of patients available for a phase 1 to 3 trial in a short period of time. This is possibly not going to happen in oncology, but there are many things that people hope for in terms of regulatory approvals in 2 weeks rather than in many months, and many other aspects. So what creative solutions have you found for your challenges? Who would like to volunteer?
Yeah, perhaps I can start. We have, in February last year, we realized we had a drug to treat COVID-19, and this was used originally for respiratory diseases, but we managed to fiddle around and during organoid trials and realize that COVID-19 is the perfect recipient for our drug. So immediately we had to sort of invent the trial as we went along, because no one really knew how many deaths would occur. And if I remember the news in March 2020, where everyone was saying we're all going to die, and this sort of scenario of everybody falling over in the streets, and then all of a sudden we had to set up a trial based on this assumption, and in the end the pandemic didn't occur as everybody thought it would do. So those who set up proper endpoints in these trials were very lucky, because in the end it's all about success in a trial, reaching the primary endpoints, and not just fiddling around. It's a matter of time. As you can see, the emergency use applications were mostly successful, in particular for drugs which had nothing to do with COVID-19. So the recommendation from our side, having gone through this, is to set as broad a trial, broad as endpoints as possible, to enable you actually to meet more endpoints than you actually plan to do.
This probably pans a little bit what was discussed at the beginning, the importance of adaptive trials and really think creatively about the endpoints as we go forward. So if I can just go through the panel and ask everybody to just give us the 1 or 2 lines, less than a minute, about what do you see is going to be the most important advance in development strategies in oncology? Can I ask Aram to start?
Sure. Well, I think, and I hope that we'll get a better understanding of the tumor microenvironment, and that we'll be able to adapt things like imaging, diagnostics, and other endpoints to the mechanistics of these approaches.
Yes. So the mechanism of the tumor microenvironment. Yes.
And the action on the tumor. I mean, one example with glioblastoma, we think we're going to block repair of blood vessels, so we look at perfusion as one of the endpoints. So this is the type of thing that, that's a very basic example, but I think more of that sort of thing, I hope, will be the norm, instead of simply adhering to what everybody else does.
Great. Jürgen, your 2 minutes, your 2 seconds.
Yeah, on the regulatory side, more openness to scientific measurements, to surrogate endpoints that have a scientific founding, but which are not really required to be well known since years. What else? Yeah, I agree with the tumor microenvironment. If I would one wish, I would also that's a wish for a long time, and I think it will never come true. But better animal models, better predictors that help us to translate, that give us a better understanding how we translate the preclinical into the clinical situation. It might well with ex vivo studies where we can use tumor samples from patients and better understand how we can treat based on the treatment, on the learning from these tumor samples, 3D cell cultures or whatsoever, to have a better transition into the clinic.
Yes, so real translational models. That's everybody's hope.
Yeah,
Peter,
that's what I've said. Yeah, everybody hopes for it. But
absolutely. No. Peter.
Sure. As this is an immuno-oncology panel, I would certainly say targeted and personalized cancer therapies will need to be focused on. And we're not just talking about innovation, we're talking about a lot of things which are going on and need to be trialed and tested and also shown to be effective.
Very good. Richard.
Thank you. Well, of course, I agree with most of the comments that have been made, targeted, effective medicines. But I think the overarching thing that we probably learned in the last 12, 18 months is the power and the value of communication, particularly communicating what we're trying to achieve and why. I think it's the why that's so important, that we must try and compel that to the regulators, to the investigators, and to the patients. And with that powerful communication, hopefully we can get compliance and meaningful results. So I probably just wanted to leave it there, if I may.
Right, Richard. So change management starts with a meaningful why and making people want to change. Scott?
Mine, very quickly, mine is further understanding of primary resistance in immuno-oncology and secondary resistance and how we overcome that. And then second is going to be a better understanding on the immune stimulatory response of the immunotherapies and how we can further take advantage of that in the treatment of cancer.
Thank you very much, Scott, and all the panelists today, and also with my co-chair, Matthias. I would like to really appreciate the many different perspectives that you have brought today that really focus on what are we going to do to demonstrate efficacy and really have patients benefit, not just tick a box and get approval based on box ticking, but really patient-driven, science-driven clinical development that provides benefit for the patients. And there has been a lot of practical advice and a lot of experience here. So really, thank you very much for a most enlightening and entertaining panel discussion.
Thank you.
Thanks.